All right, so this week we were trying to identify a certain type of behavior in mice, which we call wall-seeking behavior. So the premise behind this is that as a mouse is traveling through a field or a forest, it's going to try and avoid these open areas and try and find as much cover as possible because they're the ones that get eaten. So they're basically the chicken nuggets of the forest. So they get picked off, and in order to pass on their genes to the next generation, they want to be much more successful. So in order to be much more successful, they have to basically be hiding or trying to find cover when they're in an open area, so that way they're not ambushed or picked off by a predator. So to study this, we borrowed some lab mice from Noah Ashley's lab over in EBS. And then we put them through a series of arenas. So each arena had differing amounts of cover. So we had an arena with walls, so it had 4 walls on every side. We had an arena with no walls, which we called an edged arena. And then we had an arena with a combination of both. So it had 2 walls on 2 sides, and it had 2 edges. So half of it was basically covered in walls, and the other half was just uncovered. In order to track or quantify movement a little bit easier, what we did is we broke these arenas down into these 8x8 grids. So there's a total of 64 squares, and we would record the number of times that a mouse would be in a certain square at a certain time. So what we were doing is we were comparing a null and an alternate hypothesis. So the null hypothesis for the study was that the mice don't have a preference for cover. They're not looking for a certain type of cover. They're not looking for walls. They're not looking for edges. They're not looking for centerpieces. They just really don't care, and they're just kind of moving randomly throughout their habitat. The alternate hypothesis for our study was that they are looking for a specific type of cover. They're looking specifically for either walls or edges, and they're trying to avoid staying out in the open. So we went through our experiment. We spent about 10 minutes at an arena, and then within those 10 minutes, every 15 seconds, we were marking down where that mouse was, whether it was in a wall cell, a center cell, or a edge cell. So when we had our data, what we did was we compared our data to basically a null hypothesis. So if this mouse was just kind of randomly walking throughout its habitat, it's going to be crossing through each of those cells at a fairly equal rate. And so what we did first was we calculated the expected values. So if this mouse is randomly walking around and it's not choosing between walls or edges or centerpieces, each one of those cells in that 64 cell grid has an equal chance of being selected for. So in order to kind of mathematically represent that, we took the proportion of the values. So what we did was take x divided by 64. So we basically took the number of wall cells or edge cells or center cells, and we divided that by the total number of cells, which was 64. Then we multiplied that, we multiplied that by the number of observations that we made. And so the number of observations was 40. And so I don't know how well you can see this on that screen. A bunch of crappy markers. So we took the number of observations, and then we just multiplied 40 by the proportion of each type of square. So we did this for the number of wall squares, edge squares, and center squares. So when you go through this and you determine the number, the expected values for each of these arenas, for each of these values, you get some pretty plain data where these mice are just kind of moving around, kind of ambling. So for the first one, with walls, you have a mouse that's just kind of moving around. And it's not, specifically trying to target walls or edges or whatever. So in our 4-walled arena, so we had an arena with 4 walls. The mice had a 0% chance, basically, of being seen in a edge an edge cell. These other two groups, the walls and the centers, were the only two categories that we were comparing here. So we took the proportion of the number of edge cells and the proportion of the number of wall cells, and then we multiplied by the number of observations to get the what would be expected in terms of observations for each of these values. So we have 17.5 observations if this mouse is just randomly moving, and then 22.5. So this math is really easy here. So we've already calculated the percentages for each one of these groups. So when you have 4 edges and 4 walls, so yeah, I guess you have the exact same proportion between groups. So you have 28 edge cells, you have 28 wall cells, so you have the exact same proportion. When we're calculating proportion for our arena that had basically 2 walls on one side and 2 edges on the other, we can do this one of 2 ways. We can take our value 17.5 and we can divide it by 2, or we can take 14 divided by 64 and then multiply it by the number of observations. Either way, you get the same thing, which is roughly 8.75. And then the number of center cells doesn't change per arena that we compare. So that's 22.5 again. So now that we have our expected values, we went ahead and gathered some data. So once again, we watched these mice for 10 minutes, and every 15 seconds we marked down where they were on this 8x8 grid. So we then tried to make as much data as possible. So I had the students go through, or you all, not the students, but I had you all go through those arenas to try and get as much data as possible and have you all average them out. So just some random data that we can throw up here. You could say you have, I don't know, what, 35, 0, 0, 5, I don't know, 30, 15. And then for our 2 edges, 2 walls, we could say, I don't know, 32, 6, and 2. So now that we have, that's terrible too. A little bit less worse. But now that we have our data, we can go through and actually calculate a chi-squared value for these data. So I'll make another chart. And so I'll be going through and I'll be walking you all through this for each one of these cells. So we had 4 walls right here. And we're going to put those values into our chi-squared equation right there. So right here we have the sum of the observed values minus the expected values squared, and then divided by the expected values. So what this is doing is it's basically exaggerating differences between values, and then making it proportional, basically across all the observations we've made. So if you have a observation that's really, really close to an expected value, you'll have a very small numerator, and then when you divide it by the expected value again, you just kind of minimize it much more. So this is basically penalizing values that are really closely related to each other. So if you have an observed value and an expected value that are really close together, you'll get a really small chi-squared value. If your observed value is different from your expected value by a lot, this squared value, or this exponential right here, basically makes sure that it's being exaggerated to a certain point and then being compared to the expected values. This squared function also makes sure that everything here is positive. So instead of just adding up positive or negatives and then getting like 0, we're going to be adding all these values up, so that way we can have some sort of cumulative effect and then measure the magnitude of differences between these 2 data sets. So for the first one, we observed 35 during basically our data collection stage. We observed the mouse 35 times in a wall square in our 4-walled arena. So we'll do 35 minus 17.5. Write it. We'll square that. So this is going to be the numerator. Excuse me. The numerator for this formula. Then we'll divide it by 17.5. So this equals 17.5. So we just write 17.5 for chi-square value. And then we do this for every single one of these cells. So what we're doing is we're just matching up each of these cells to 0, 0, 0, 0. So we have zeros, and then what we're doing is we're just filling in for this equation inner blanks. So for the number of center cells that we observed in our four-wall arena, we observed 5. We're going to take 5 minus 22.5 right here. We're going to square that, and then we're going to divide that by 22.5, and that will give us a chi-squared value of 13.61. I hope you guys don't think I'm doing this in my head, because I'm not. I have the answers right here. Yeah, so for our next column, we'll take 30 minus 17.5, square it, and then we'll divide by 17.5 again, and that'll give us 8.93. We'll take our center cells for our four-edged arena. So we have 15 observed times, or 15 observations. We'll take 15 minus 22.5, we'll square that, and then we'll divide by 22.5. And so this will give us 6.94. So fill that in, 6.94. And we'll do this for the last arena. So this is our two-walled, two-edged arena. So we'll take whatever value we observed, so 32, we'll subtract 8.75, we'll square that numerator, and then we'll divide by 8.75 again. And so that gives us 61.78. We do the same thing, so take our observed value, so 6 observations minus 8.75 expected observations. So we're going to square it, and then we're going to divide by 8.75, so the expected value again. So that gives us 0.86. And then we do it for our last one. So we have 2 observations minus 22.5 expected observations. We square that, and then we divide by 22.5, which gives us 18.68. So we are more than halfway done with our chi-squared value. So the last thing we need to do is basically just add all these values up together. So this is what this symbol right here means. I forget what it's called. I don't know, the capital E, the enigma or something, I don't know. But this symbol just means basically the sum of. So we're taking all these cells and we're going to be adding them up. So when we do that, we get a value of 128.3. And so that is our chi-square value that we calculated from our observed data when we were comparing it to our expected values. So the last thing I had you all do was calculate what we call degrees of freedom. So degrees of freedom will equal, I don't know how far you can see it, and it's about there. All right, so number rows minus 1 multiplied by the number of columns minus 1. So we have 3 rows, 3 columns. So 3 minus 1 is 2. And then 3 minus 1 again is 2. We multiply those two together and we get 4. So we have 4 degrees of freedom. Let me erase this out of the way. So with 4 degrees of freedom, we would then go on our critical values chart and then try and identify which chi-square value led to a certain p-value. At the very beginning, we had a specified p-value of 0.05, so that way we weren't changing our data at the very end to match some significant level. So yeah, we had some very significant results. So when we plot this in like a normal distribution curve, so if we have this experiment run several million times, or just a couple hundred times for that matter, you end up getting basically a normal distribution of differences between those two values, or between these two groups. And so if you wanted to plot our data that we observed here, we're like, I don't know, like right out here. So this is an extremely significant chi-square value. So this wouldn't happen by chance. It's much, or way too significant to have occurred randomly. So with that, we can conclude that yes, lab mice do have some sort of instinctive behavior where they're actively preferring or seeking out walls or some sort of cover. So if we want to look at this in a little bit more detail, we can look at the observed values, or basically the chi-square formula between the direct comparison between these two categories. So our two edges and two walled arena was a direct comparison between the two categories, walls and edges. And so if we look at the chi-square values right here, so we have an extremely high difference, 61.78 for a number of walls, and 0.86 for the number of edges. So just looking at these two and preemptively just comparing the two without going through any other statistics, you can say that we observed a much greater amount of wall cells, of that mouse being in a wall cell, than we would have expected if that mouse was just randomly moving around. And so that difference was not the same with edge cells. So this mouse is actively seeking out walls, and that was the point of all this math. So with that, we can conclude that mice do have some sort of wall-seeking behavior, and it is instinctive. So even mice that have grown up in a lab and they've never experienced some sort of predator will have a wall-seeking tendency that's basically been passed on from generation to generation. So one thing that I did ask early on in each of the labs was about either proximal causes or ultimate causes for this behavior. And so a proximal cause is some sort of environmental stimuli that's happening immediately on an individual. So in this case, having some sort of predator, some sort of big, scary, I don't know, human-looking thing that's staring at them, causes them to look for some sort of cover or hide, or try and find a place to hide. The ultimate causation is basically how that behavior is affecting gene flow later on in life, and how it's going to affect successive generations after this. So in the case of these mice, the ones that are actively seeking cover, their genes are being passed on to the next generation. The ones that are just being free-spirited and are hanging out in the middle of an open field, they'll get picked off by like a hawk or an eagle or an owl. And so their genes, if they haven't had a chance to reproduce yet, their genes are basically removed from that gene pool for forever. So that's how basically behavior is evolving. So you have basically some sort of proximal cause that is affecting the short-term behavior of this organism, and then you have the ultimate cause, which is affecting the long-term behavior of this organism. So, yeah, that was about it for this week's lab. Next week is all about pig dissection, and so I'll see you then.